blood glucose level is maintained within physiological limits, 60 to 100 mg per deciliter, that is, true glucose, in fasting state, and 100 to 140 mg per deciliter, following ingestion of a carbohydrate-containing meal, by a balance between two sets of factors. Rate of glucose entrance into the bloodstream. Rate of its removal from the bloodstream. Rate of supply of glucose to blood. The blood glucose mainly may be derived directly from the following sources. By absorption from the intestine. Breakdown of glycogen of liver, hepatic glycogenolysis. By gluconeogenesis in liver, source being glucogenic amino acids, lactate and pyruvate, glycerol and propionyl-CoA. Glucose obtained from other carbohydrates, for example, fructose, galactose. Rate of removal of glucose from blood. Oxidation of glucose by the tissues to supply energy, that is glycolysis. Glycolysis is basically the oxidation or conversion of glucose to pyruvate and lactate to produce energy in the form of ATP. Glycogen formation from glucose in liver, that is, hepatic glycogenesis. Glycogen formation from glucose in muscles, that is, muscle glycogenesis. Conversion of glucose to fats, that is, lipogenesis, especially in adipose tissue. Synthesis of compounds containing carbohydrates. Blood glucose is utilized, for example. Formation of fructose in seminal fluid, a very important point to be remembered, for laboratory point of view. Formation of lactose, sugar of milk, in lactating mammary gland. Lactose is the sugar, which is present in milk. Synthesis of glycoproteins and glycolipids. These glycoproteins and glycolipids are present in cell membranes. Formation of ribose sugars from glucose, required for nucleic acid synthesis. For example, for the synthesis of DNA and RNA. Excretion of glucose in urine, which is called glycosuria. Glycosuria appears when blood glucose level exceeds the renal threshold, that is almost 180 mg per deciliter. All of the above processes are under substrate, end product, nervous and hormonal control. Condition of blood glucose in post-absorptive state. What is postprandial state? It is a state or condition, following ingestion of food, is called postprandial state. Absorbed monosaccharides, glucose, fructose or galactose, are utilized for oxidation to provide energy. Remaining in excess is stored as glycogen in liver and muscles. In well-nourished individuals, glycogen stores are fairly saturated. About 40% of absorbed glucose is used for lipogenesis. Some are used for synthesis of glycoproteins, glycolipids. The above store of liver glycogen can supply for 8 hours, approximately as per rate of utilization stated above. Muscle glycogen store is approximate 0.7% of the weight of muscle mass. If the muscle mass of an adult is approximate 35 kilograms, the muscle glycogen amounts to 245 gm. Muscle glycogen cannot provide blood glucose by glycogenolysis due to lack of the enzyme glucose 6-phosphatase. Glucose 6-phosphatase enzyme is present in liver, but not in muscles. End product from muscle glycogen is transported to liver, where it is finally metabolized by glucose 6-phosphatase, which is only present in liver. It can supply indirectly by gluconeogenesis from the pyruvates and lactates, the products of muscle glycogenolysis. Thus muscle glycogen can provide indirectly blood glucose approximate less than 25 hours. When blood glucose rises more than 160 to 180 mg per deciliter, that is the renal threshold, glucose appears in urine, glycosuria. This is an abnormal state. In normal intestinal absorption, such situation does not occur. It can take place within foreload or disease processes. Autoregulation, fundamental regulatory mechanisms. Process of hepatic glycogenesis, glycogenolysis, and tissue utilization of glucose are sensitive to relatively slight deviation from the normal blood sugar concentration. As blood sugar tends to increase, glycogenesis is accelerated. Utilization of glucose by tissues is increased, resulting to fall in blood glucose level. The reverse occurs as the blood glucose level tends to fall. Normal balance between production and utilization of blood glucose at a mean level of circulating glucose of approximately 80 mg%. This level of sensitivity is determined, to a considerable extent, by the balance between insulin in one hand, hormones of adrenal cortex and anterior pituitary on the other hand. Overall effect of insulin is to lower the blood glucose level and adrenocortical and growth hormone to raise it. Inasmuch as these two sets of factors are mutually antagonistic to each other, the processes of hepatic glycogenesis, glycogenolysis, and glucose utilization, and also the blood glucose concentration, are continually exposed to fluctuating influences under physiological conditions. These include absorption of glucose from intestine, physical and mental activity, emotional states, etc. Primary effect of majority of these is to cause a rise in blood glucose. This results in decrease in delivery of glucose by liver, acceleration of utilization by tissues, simultaneous increase in insulin secretion, stimulated by elevated blood glucose concentration, results in increased hepatic glycogenesis, decreased gluconeogenesis, decreased output of glucose from liver, increased utilization of glucose. As a result of above, the blood glucose concentration tends to fall. As blood sugar tends to decrease, a drop in blood glucose concentration below the normal resting level causes decrease in secretion of insulin, resulting to decrease in ratio of insulin/glucocorticoids and growth hormone decreases. Increased production of blood glucose, mainly by gluconeogenesis. Decreased glucose utilization. Due to the above actions, blood glucose tends to rise. If the blood glucose falls below to hypoglycemic levels, additional emergency mechanisms come into play. Stimulation of secretion of catecholamines by hypoglycemia, resulting in hepatic glycogenolysis and rise in blood glucose. The increase in catecholamines may also stimulate production of ACTH, hence of adrenocortical hormones, causing increased gluconeogenesis. The blood glucose concentration in normal health regulates itself. Efficient operation of this autoregulation at physiological levels, however, requires a normal balance between insulin and the carbohydrate active, adrenocorticoids, cortisol, aldosterone, androgens, estrogen, and anterior pituitary hormones, GH. This constitutes the autoregulation or central regulatory mechanism.